Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine this. You wake up and the first thing you think about isn't coffee, it's math. Calculating carbs, insulin doses, knowing your body's basically missing a key piece of machinery.
Yeah, it's relentless.
For, what, over 8 million people with type 1 diabetes? That's just everyday life. Always watching that blood sugar, always worried about it crashing. Especially scary if you can't even feel it dropping.
That's a really crucial point. The impaired hypoglycemia awareness. Even with all the tech we have now, pumps, monitors hitting those stable blood sugar targets it's still incredibly tough for so many.
Right. It leaves people open to really dangerous situations day in, day out.
And the long-term complications too, of course.
So that's the baseline struggle. But today we're diving into something that might change that picture fundamentally.
A potential shift, yeah.
We're looking at a new therapy. It's called Zimislecel. It's an allogeneic stem cell-derived islet therapy. Bit of a mouthful. But the recent trial results.
Wow. They're suggesting this could be like a whole new way to think about managing type 1 diabetes, maybe even restoring function.
Exactly. So our plan for this deep dive is really to unpack what's going on here. We've got the news reports, but more importantly, the actual scientific paper from the New England Journal of Medicine.
Yeah, the gold standard.
Right. So we want to pull out the key findings, understand, what the study tried to do, how they did it, and these results, which frankly are pretty surprising.
And also the limitations. Got to talk about those.
Absolutely. Got to be balanced. And what it all means for you listening in, if you're curious about where medical science is heading.
Okay, so before we jump into Zimislecel itself, maybe just a quick refresher. Type 1 diabetes, what's actually happening in the body?
Sure. So fundamentally, it's an autoimmune thing. Your immune system gets confused, basically, and attacks the beta cells in your pancreas.
The ones that make insulin. The very ones. So it wipes them out. And without those cells, your body just loses its natural thermostat for controlling blood sugar.
Yeah.
The whole system goes offline.
Got it. And even with those advanced gadgets, the monitors and pumps you mentioned.
Yeah, even with those reaching the recommended blood sugar goals consistently, it's a huge challenge. And for people who have that impaired awareness, who don't feel the lows coming on,
the danger zone is just right there all the time.
Exactly. It makes those potentially life-threatening lows, those severe hypoglycemic events, a constant risk. It's not just feeling a bit shaky. It can be much, much worse.
So, okay, that's the problem. What about solutions up till now? You hear about transplants sometimes.
Right, you do. Transplants of pancreatic islets, the clusters of cells containing the beta cells, or even whole pancreas transplants. They can work. They can restore that natural glucose control.
But there's always a but.
There's a big but, the availability. There just aren't enough donor pancreases or islets to go around. It's a major bottleneck.
So not really scalable.
Not at all. Often you need cells from multiple donors just for one recipient. It's complex, limited. It highlights why there's been such an urgent push for something new, something you can scale.
Which brings us back to Zimislecel. What is it, and why is it potentially different?
Okay, so Zimislecel, it's an allogeneic stem cell-derived islet cell therapy. Now, that word allogeneic, that's the key. Meaning the cells come from a different source, not the patient's own body. They're grown from stem cells in a lab.
Ah, so you're not limited by organ donors anymore.
Precisely. That's the potential game changer. It opens up the possibility of creating a, theoretically inexhaustible supply of these insulin-producing cells.
Yeah.
Consistent, available.
Okay, that's a fascinating idea, growing replacement parts, essentially, from pluripotent stem cells. How does that work? What's the thinking behind how they restore function?
So the basic hypothesis is this. You take these pluripotent stem cells, cells that can become many different types of cells, and you guide them, you differentiate them in the lab to become fully working pancreatic islet cells.
Like coaxing them into the right job.
Kind of, yeah. Then the idea is you infuse these lab-grown cells into the patient. The hope is they'll take root, mature, and start doing what the original beta cells did, produce insulin and regulate blood sugar naturally.
Giving the body back the control it lost.
That's the aim. Restoring that lost physiological function.
Sounds almost like science fiction, but they actually tested this.
They did. That's the VX-880-101 forward study. It was a phase 12 clinical trial, so still relatively early stages, safety and initial efficacy focused.
Led by researchers where? And who funded it?
University of Toronto researchers were key players, and it was funded by Vertex Pharmaceuticals. And like we said, published in the New England Journal of Medicine, which gives it that scientific credibility.
Right. So who are the volunteers? Who signed up for this?
They enrolled 14 adults, aged 18 to 65, all with established type 1 diabetes. But critically, they all had two specific things in common.
Which were?
One, that impaired hypoglycemia awareness we talked about. They couldn't reliably sense lows. And two, they'd had at least two severe hypoglycemic events in the past year, despite getting good standard diabetes care.
So these were people really struggling with the most dangerous aspect of the condition.
Exactly. People for whom the current best just wasn't enough to prevent these emergencies.
How did they actually give them the therapy? Was it a pill? An injection?
No, it was an infusion. A single infusion, actually, using gravity to help deliver the Zimislecel cells directly into the portal vein.
Portal vein that goes to the liver.
That's right. The idea is the liver provides a good environment for these new islet cells to engraft, to kind of set up shop and start working.
And they needed immunosuppression too, right? To stop the body rejecting the new cells.
Yes, absolutely crucial. But, and this is important, they use a specific glucocorticoid-free immunosuppression regimen.
Why is that significant?
Because standard glucocorticoids, like prednisone, which are often used after transplants, can actually harm islet cells and make it harder for them to work properly. So avoiding those was a key part of the strategy to give these new cells the best chance.
Okay, makes sense. So the big question, what happened? What were the results, especially for the folks who got the full dose?
Right, so 12 participants received the full intended dose, and the results were, well, pretty striking. First off, all 12 showed detectable C-peptide levels after treatment.
And C-peptide means?
C-peptide is released alongside insulin when the body makes its own. At the start, these participants had undetectable C-peptide, meaning zero natural insulin production. Seeing it reappear is clear proof that the transplanted cells had engrafted and were actually functioning, making insulin.
Wow. So literally went from zero insulin production back to making their own.
That's what the C-peptide indicates, yes. A restoration of endogenous insulin production.
That alone is huge.
Yeah.
What about those dangerous hypo events? Did it help with that?
That's perhaps the most dramatic finding. In that first year of follow-up, for those 12 full-dose participants, zero. None of them experienced a severe hypoglycemic event.
Zero, down from at least 2 each in the year before.
Correct. Complete elimination of severe hypoglycemic events in this group during the follow-up period. For people living under that constant threat, you can imagine that's life-changing.
Absolutely transformative. Okay, so severe lows were gone. What about just general day-to-day blood sugar control? Did that improve?
Significantly. All 12 hit a glycated hemoglobin, or HbA1c, level below 7%. That's the standard target recommended by diabetes organizations for good long-term control.
Okay.
And maybe even more impressively, their time spent in the target glucose range, that's between 70 and 180 milligrams per deciliter, shot up. The average time in range hit 93.3% by the end of the year. That suggests really stable, well-managed blood sugar levels most of the time.
93%. That's fantastic control. And the ultimate goal for many, I suppose. Did they still need insulin injections?
Here's another remarkable result. 10 out of the 12, that's 83%, became completely insulin independent.
No more shots. At all.
No more daily insulin injections needed for those 10 people.
Incredible. What about the other 2?
For the remaining 2, they didn't achieve full independence, but their daily insulin needs dropped dramatically by 70% for one and 36% for the other. Significantly less burden.
Was there any reason given why those 2 didn't quite get off insulin?
Yeah, the paper notes that both of those participants had received glucocorticoids for unrelated medical issues, which was off protocol for this study. And as we discussed, glucocorticoids aren't great for islet cell function or survival.
Ah, so that might explain the difference. It actually reinforces the importance of that glucocorticoid-free immunosuppression.
It seems to, yes.
Okay, results sound amazing, but we have to talk about safety. Were there side effects, adverse events?
Yes, there were. Most were reported as mild or moderate. Things like diarrhea, headache, nausea were seen. The most common serious adverse event was neutropenia.
Which is low white blood cells.
Right, a specific type of white blood cell involved in fighting infection. That occurred in 3 participants. It's a known risk with some immunosuppressants, generally manageable.
The reports also mentioned 2 deaths occurred during the study. That sounds serious.
It does, and it's crucial to be clear about the details. One participant died from cryptococcal meningitis. This happened after they received those off-protocol glucocorticoids we mentioned, which likely worsened the effects of the necessary immunosuppression, making them more vulnerable to infection.
Okay. And the second death?
The second death was due to the progression of a pre-existing neurocognitive condition the participant already had. Importantly, the study investigators concluded that neither death was considered directly related to the Zimislecel therapy itself.
But the immunosuppression was a factor in the first case.
Yes, which highlights that the immunosuppression needed to prevent rejection carries its own risks. That's always a trade-off in transplant medicine, including cell therapies like this.
So, given these really promising but obviously early results, what did the researchers themselves say? Were the limitations here? We need that scientific caution.
Absolutely, and they were very upfront about it in the paper. First, it was open label. Everyone knew who got the treatment. That can potentially introduce bias.
Right.
Second, very small number of participants. Only 14 total, 12 in the full dose analysis. You can't draw broad conclusions from such a small group.
Need bigger numbers.
Definitely. They also noted some analyses used outcomes that weren't strictly predefined in the original plan, and they explicitly state that confirming these findings requires larger studies, longer follow-up, and, a more rigorous trial design overall.
Which sounds like standard scientific process. Good to know they're aware. Are they doing more trials?
Yes, they mention a more advanced trials already underway, which is a positive sign. They're moving forward.
Okay, so pulling back a bit.
Yeah.
What are the bigger picture implications here? If these results hold up in larger trials, what could this mean?
Well, it's potentially profound. We're talking about moving beyond just managing type 1 diabetes with external insulin. This hints at actually restoring the body's own ability to control glucose physiologically.
Like fixing the underlying problem, not just treating the symptom.
Exactly. Imagine the freedom, the reduction in daily burden, no constant calculations, injections, finger pricks, the constant vigilance, and reducing that ever-present fear of severe hypos. It's a massive potential shift in quality of life.
And the stem cell angle seems really significant too, beyond just diabetes.
Oh, absolutely. This study provides really compelling human evidence that you can take pluripotent stem cells, turn them into functional pancreatic islets, put them into a person, and have them work.
So proof of concept for regenerative medicine.
A powerful one, yes. It bolsters the whole idea that we could potentially grow replacement tissues or cells for all sorts of serious diseases where cells are damaged or lost. That concept of an inexhaustible supply becomes a bit more real.
It really feels like a step towards a new kind of medicine. Not just pills or surgeries, but biological fixes.
It does edge us closer to scalable cell-based therapies for conditions we've always thought of as just chronic, lifelong management situations. It makes you rethink what a cure might even look like.
It's definitely one of those aha moments in science, even with the caveats.
For sure, a very exciting development.
So wrapping up our deep dive today, we've looked at Zimislecel, this stem cell-derived therapy for type 1 diabetes. The early results, highly promising, suggesting it might actually restore the body's own insulin production and glucose control.
Yeah, offering a potential future with drastically reduced burden, maybe even eliminating the need for insulin injections for many, though we have to stress it's still early days, small study, more research needed.
Absolutely. But the potential implications are huge, not just for diabetes, but for the whole field of stem cell research and regenerative medicine.
It really showcases the power of this technology and where it might take us in treating chronic diseases.
So here's something to think about as we finish. What does it really mean when medicine starts moving from just managing symptoms to potentially restoring fundamental biological functions? Think about other chronic conditions. Could this kind of approach work elsewhere? And how does that change our whole idea of what a cure could be?
A fascinating question. It definitely invites us to imagine what might be possible next.
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.